we don't know is how to analyze this data from now on. Right? This is the next step. How do I use these alignments in order to get read counts? I want to get read counts per gene. That's the most important part right now. We want to get a table that tells us how many reads were aligned to each of the genes in my reference genome. Now, there are two ways. As usual, there's always more than one way of doing things. And we're going to be doing this the easy, ugly, but quick way, right? The traditional way is if we had mapped our reads to a reference genome, then we would have gap alignments, like the ones we showed, right? Where some of the reads will be split across different loci because they belong to different exons, yeah, and they were aligned, so we would have to count them only once per gene. So it's kind of a, it's a bit tricky. So for this, there are some tools that have been developed. One of them, you can find it under INEA 08. You'll see one that's called here under subread 163 Linux x86-64. There's going to be a binary directory, and inside this binary directory, you'll have different commands. The command we would be using would be feature counts. So how do we execute it? We just do / the direction, and then feature counts, -- help, and you should get, if you get the help, that means that the program is working. It's in INEA 08, under subreads binary, or bin. There you will find a set of tools. One of them is called count feature, or feature counts, sorry. And this one, which has a very long help, this one will be in charge of getting an alignment file, read it line by line, extract the positions that have been coded as exons, group the read counts per gene, and then produce a table with the read count per gene per library. We can do the same. Yeah, but this is a tool. This tool is very useful when you have gap alignment, like the ones you would get if you were to map your reads to a genome assembly instead of a transcriptome assembly. But we've got a transcriptome assembly, so getting the read counts is a lot easier than that. So the main commands, the main thing that we need to take into account is, one, an annotation file. This should be an annotation in GTF format that contains information of the coordinates where the exons are, where each exon is, so that we count the reads that align to those positions, and then knowing which exon belongs to which gene, we can assign the total number of reads to each gene. The output file is just going to be a table, and the input file 2 and 3 and 4 is going to be all the alignment files that we are going to produce. So if we had used the reference genome, we would have to use the annotation file of that reference genome in order to run this tool and get the table count. But we didn't use the reference genome, we used the reference transcriptome. And that makes it easier for us. How does that make it easy for us, or easier for us? Because we can just use bash to get the information we want by knowing how the format works. We all have these SAM files, and we've all seen what a SAM file looks like. we know that one of these columns will tell me what is the transcript to which my read was aligned to. The first one is the read name, the second one is some flags that tell me things about the read, and the third one is the reference genome where it was aligned. This is the one that I need, column 3. And since I have one record per read, if I count the number of lines that have each of these names, I will have a read count per gene. Does this make sense? It doesn't make sense at all? So remember, we have one line per read. And that line describes where this read was aligned. So this line here, that starts here and finishes here, starts here and finishes here, this line is telling me that this read aligned to this reference, to this sequence in the reference, which is a gene. From this position, it had 17 of mapping quality, 100 matches, and then information about the sequence, additional information. But what I want to know is that this is the sequence it was mapped to. So if the next one, the next read, which is read 59, 502, etc., maps to the same position, that means that now I have two reads mapping to the same gene. It's a different position, but it's the same gene. Now, if I count this again, I know it's a different position, but it's the same gene. So how many reads have I already aligned to the same gene? So far here, 3. Here, 4. This is a different one. So you don't count it twice. Now, this is so that we understand. If we count the number of times this name appears in my alignment, we will know how many reads were aligned to each of these genes. Does that make sense now? All right, cool. And how do we select this column? How do we select one single column from a file? Who remembers that? How do we select one single column of a file? Cut. Cut minus F. The number. Which number would it be? 3, exactly. If we use cut minus F3, then we will be selecting only the third column of my entire alignment file. So let's just play around with it a little bit. Why is that? Because the headers, the ones that have the at the beginning, since they don't have several columns, we're just selecting the first one, or the second one in this case, the last column that was available. But that doesn't matter, we can get rid of that later. So, what do we do with this? We need to find a way of counting it. And for that, we already have a set of tools. We have already done this before. The first thing we need to do is sort it. Sort it alphabetically, so that all the occurrences of the same gene are following each other. So we cut the third column, we sort it, and then we use the command called unique. Unique. Unique, what it's going to do, it's going to see all the lines that are the same. In this case, all the names that are the same. For example, this 81G2109 that appears 5 times in a row. Here, 1, 2, 3, 4, 5. Or 4, sorry. Yeah, they're going to be compressed into a single line. But since I want to count it, I just add this flag that says count, minus C count. And now I just want to see what the output would look like. So for each gene, we're getting a count. That is, this count corresponds to the number of lines in the file that have that gene in the third column. So this is going to be useful. Now, instead of head, we're just going to use, we're going to clean it up a little bit. This is just a quick way to eliminate the leading spaces. So the last one, so far until unique C, this is something you're used to. You've already seen all this. These set commands, these are two set commands, the first one and the second one. These are just saying, if you have leading spaces, yeah, remove them, I don't want them. Yes, this is what the first one does. The second one says, if you have any space, which is the one between the number and the name, yeah, replace it for a tab. And that's it. That's all the command is doing. We just press enter, and we just got our gene count. The number, we already have a count of how many reads there are per each transcript. If we want to take a look at it, you'll see that this is here. Compare the size of this file, 53 kilobases, to the actual alignment file, which was, where are you? Where are you? Here it is, 40 megabases. So there are a few things that we need to get rid of, certainly, but this is easy to get rid of. Yeah, the important thing is that you can see that for all the genes, for every gene you have a count, a number of reads that have been aligned to that gene and that you have finally counted. Okay. Please be aware of the quotes. If you open and you don't close the quotes, this is not going to work. You need to open and close the quote. After the set minus E, there is open quotes and close quotes. Just be mindful of that. The second one has the same issue. Open and close quotes. Now, do you remember that you have to do you do understand that you have to do the same thing for each of your files, right? For each of your SAM files. So what would be the fastest way to do it, just like we did with the previous one? You create your bash file, copy your lines, replace what you need to replace, and then just execute your bash file. So just like before. Yeah. So you just create your bash script, repeat the same command, just change the name of your SAM file and the name of the output file. So you just create your bash file. Now you just save it, right? And now we can execute this bash file. It's going to take a little while. going to take just a little while. Yeah, but you're going to generate one gene count per sample. And now that we have all these gene counts, it's time to merge them into a single table. Now, first thing, let's make sure that all the files have the same number of lines. Have we all produced this? Is anybody having trouble? It's okay? Good? How are you doing, guys? Working? And now, R. Finally, we want to merge all these tables into one single table. And we want to write it. And we want to know that each column means it's a different sample. Right? We want to generate a count table that comprises the information of all 6 libraries in a single table. That's what we want to do. Right? And for that, we're going to merge all these tables. But we have to make sure at the time of merging that the reads for each of the gene correspond to the same line or to the same row in the table. That is, for libraries 1, 2, 3, 4, 5, and 6, gene 1 will have the count in the right position. So for that, what we're going to do is use the command merge in R. So now we go to R. Just capital R, enter, and you will be in R. If you want to use RStudio, it's fine. You can use our command line R. It should be fine as well. Yes. Sí, no correcto, ¿eh? Bash. Oh, if you haven't run your bash script, run it so that you need to get one table per sample with your read counts per gene. So the first thing we do in R is to load our tables into memory, right? We need to create objects that store the data of our tables. How do we do that? Hey guys, how do we do that? How do we load our tables into R? What command do we use to load our table? To read tables and assign them to variables in R. Who remembers? Read dot table. So sample 1 is going to be read table, the name of my table, which should be 01 dot gene count CSV. The separator should be tab, because I made sure that there was tab, and the header should be non-existent. There we go. Oh. We can use it in OS2. We can use either of those commands. Either read.table, where we have to specify what is the separation between the columns, or the read.delim, where we don't have to specify the separation, because it assumes that it's tab separated. Yes? don't worry. Yep, it's normal. Yes. Good. Enter. Permission. Ah. Yeah, that's right. This is the reason why we always say don't use spaces in Linux, because Vim está editando dos archivos, está editando count y reads. Son dos archivos diferentes. Por eso te dice two files to edit. Y por eso cuando cerramos uno, el otro nos dijo, oye, todavía estás editando un archivo más. ¿Ya? Pero acá está count. ¿Ya? Que tiene la información que necesitas. ¿Ya? Solamente tienes que cambiarle de nombre a count.sh. ¿Ya? No tienes que, pero es mejor. Y ahora es bash count.sh. Yes, now do the same thing for S3, S4, S5, and S6. So if you want to see the list of all the files you have created and you have read, you just do ls parentheses, parentheses, enter, and you see that you have created S1, S2, S3, S4, S5, and S6. Each of these files, or each of these variables, is storing the information of one of the files that you generated. So yeah. That's done? Yep. I know this is very labor intensive because you have to write each line every single time. If we were to use the tools that we've already learned so far, for loops, we could do all of this in just 2 or 3 lines and just repeat it. Yeah? But we're just doing it by hand. There are not that many lines to it. If we had like 100 files, yeah, probably it would be better to use a for loop. But we only have 6. Which one? Yeah, we're going to get rid of those. Don't worry. Yep. Oh! That is a good catch. That is a good catch. Oh. Come on. Yeah, you're absolutely right. Sorry. It's the other way around. The first column is the count, the second column is the gene ID. And now that we have these tables, we can use, and that we have renamed the column names. Yeah, that's what we use this command for. Names of each of the tables into count and gene ID. Why? Because we want to merge them using those names. So for that, we'll create a new table. It's going to be called gene counts. Sorry, I promise this is the last time I change my mind about something. Okay. So now comes the interesting part. We have the command merge. Yeah, and if you don't know what the command merge does, in R, you simply put a interrogation sign at the beginning. And say merge, and say merge 2 data frames by common columns or row names. Or do other versions of database join operations. And here it says, you require an X, you require a Y, which are two data frames. So the tables that we just created. Yeah. And then we said by which column do we merge it. And that's why in all our tables, we have created one column with the same name, gene ID. That's what we have done. We want to merge all our tables using gene ID. But unfortunately, we have to do it one table at a time. So we use merge. The first table is going to be S1. The second table is going to be S2. And we're going to merge it by gene ID. Where are we going to store it? In another table called gene count. Oh. There we go. And now, when we see what our gene count looks like, we'll see that we have managed to merge both tables into a single one, where they have a common gene ID, and for each sample we have stored the count that we got from each of the alignment files. Now, you may be asking, how do we merge the third one? Merge. Now we're going to merge my gene count with S3 by gene ID, and we're going to write it all the way back to gene count. So we're going to overwrite gene count with the product of merging the previous gene count and the S3. And then we're going to do that the same with S4, and then with S5, and then with S6. So basically we've used the command merge. What we've done is use the command merge to merge several tables into a single one. And we've kept the order of the count in each table following the gene ID column. Why? Because all the tables that we created, they had the same gene ID column. If, this is an important thing about the merge, if one of the tables you wanted to merge didn't have one of the gene IDs, you will still write the gene ID, but the count will be either non-applicable or 0. You will coerce that gene that wasn't present in some of your samples. And now this is what we call a gene count table. The only thing we need to do now is get rid of those headers. It's all the lines that have period. LN period, SO period. I am overwriting it. That's the whole idea. This is one of the features of R. When you overwrite something, and in the operation you use the same content, what you're doing is basically updating your table. You're saying, use the results from my previous table, which are still here, add this, and store it here again. It's not deleting anything. As you can see, each sample has been added sequentially. So, yeah, it is actually, internally what it's doing, it's creating a temporary copy, which is going to be your new copy. Then you're using the current copy to duplicate and to generate the whole thing. Then you're creating this temporary copy, will have all the new information, and you're deleting the old copy and moving this temporary copy into the new copy. That's what internal is doing. That's a good thing about interpreters, that they do all this number of steps. They do it without us seeing them. Because if we had to put one command for every single step of that, we would go crazy. Yeah, please be aware that the merge function only accepts 2 tables at a time. That's why we haven't used S1, S2, S3, S4, S5, and S6 in a single go. It's because you can only merge 2 tables at a time. Wouldn't be pretty. It wouldn't be pretty. There are always ways to do it faster, but not necessarily clearly. Actually, I think this looks pretty nice for a differential gene expression thing. Look, the first 3 samples, they all have, for gene 1, they all have around 1,500 reads. Whereas for sample 4, 5, and 6, they all have around 400, which is almost a 4-fold change. Here you have nearly 3,000, and here you have around 700, which is roughly 4 times fold change. And that's the same for most of these genes. No, you do need to do statistical. Just because I see it by eye, yeah, and yeah, this is pretty obvious actually. When you look at it, it's pretty obvious. Just because it seems to be pretty obvious doesn't mean that I don't have to run a statistical test to prove it. Every single journal is going to ask me, if you say something is significant, you need to show a statistical test that proves it is statistically significant. Otherwise, they won't accept it. Now, be aware, I didn't explain this before. This experiment, yeah, first of all, this is just simulated data, but this is something you would kind of expect in the real world as well. Yeah, this belongs to 3 repeats and 2 treatments. So we have a control in the first 3 samples and a test or a test 1 or a environment 1 in the next 3. And we've done 3 repeats. Why do we do 3 repeats? For statistical purposes. So now, if we wanted to save this file, to save this table as a table, we would use a very, very weird command in R that is called write table. Write table is what we'll use to write this table down. How do we use it? Simple. The first argument is going to be what table do we want to save? It's going to be what's the name of the file we want to save it in? We want to save it in genecounts.csv. What kind of separator we want in the text file? We want to use tabulation, tab. Do we want the data to be quoted or not? No, I don't want quotes around my data. Do I want the raw names, which is this number here, 1, 2, 3, 4, 5, 6, to be written in that file? No, I don't want those raw names to be written. Yeah. Don't worry, we will get to that. I'm just teaching you right now how to save the data. Because this read count data is going to be useful for pretty much anything we want to go as a downstream analysis. Differential gene expression, network analysis, all these different things. So for example, if you see, I've already created my gene counts. And if I do a head of my gene counts, I'll see that I have 7 columns, gene ID, sample 1, sample 2, sample 3, sample 4, sample 5, sample 6. ¿Verdad? Sí, si no te funciona algo, tienes que decirme. Porque todos tenemos que dar la misma información, si no. ¿Lo volviste a escribir de cero? Yeah, you need to re-generate that. You need to rerun the gene count from the sum, yeah, and reload all the data. You should have S4 complete now. You should. So we've written this. We have the table, we have the gene count table. That is excellent, sorry. Okay, so we'll need to go for a quick break before the food finishes. Please, we only have 10 minutes this time. So, hurry, we are in a rush. So, please, go take a break, eat something, drink something, rest a little bit, and I'll wait back. That looks beautiful.